In the next section, we'll describe the physiology of smooth muscle. In this section, we'll discuss smooth muscle. There are multiple differences between smooth muscle and skeletal muscle. One major difference is the high prevalence of gap junctions in smooth muscle. Smooth muscles can be separated into categories of single unit versus multi-unit smooth muscles, depending on the frequency with which you find gap junctions in that smooth muscle. Smooth muscle which has many gap junctions, which would suggest that that smooth muscle operates as a single unit, are categorized as single unit smooth muscles. One example of this would be the GI tract. In the GI tract, you have coordinated contraction of many smooth muscle cells, and this is referred to as a single unit smooth muscle. Multi-unit smooth muscles have sparse gap junctions, and therefore each individual muscle cell has its own innervation and therefore its own control. An example of this would be the iris. Here you have individual control of individual muscle cells, and therefore you have sparse gap junctions, and you don't have the type of coordinated activity as you would find in the GI tract. Another major difference between smooth and skeletal muscle is the nature of the innervation of that muscle. In skeletal muscle, you have the neuromuscular junction. In smooth muscles, we have varicosities. Varicosities are sections of axons that have gaps in the myelin sheath, and the gaps are surrounding swellings in the axons, and these swellings have vesicles, and these vesicles allow exocytosis of neurotransmitter. But you don't have the type of specialized structures that you find in the neuromuscular junctions. So these varicosities function as the motor end plates. The next slide illustrates some of the specializations in smooth muscles. So at the top we have a postganglionic autonomic neuron. This could be releasing norepinephrine or acetylcholine. We find a series of varicosities along the length of this axon rather than a terminal distinct neuromuscular junction. On the smooth muscles themselves, we have multiple gap junctions. We do see actin and myosin, and they do overlap to facilitate contraction, which we'll discuss shortly. But what you don't have are sarcomeres. You don't have the regular linear arrangement of these molecules that are found in sarcomeres. Another major difference is the source of calcium. While there is a sarcoplasmic reticulum in smooth muscle, the sarcoplasmic reticulum is much less well developed when compared to skeletal muscle. Instead, caveola, which are invaginations of the membrane, surround locally high concentrations of calcium, and here is a major source of calcium for contraction of smooth muscles. The next slide illustrates a typical single unit smooth muscle from the small intestine. So we have a cross section of the small intestine. We have an axon with multiple varicosities along the intersection of this muscle, and we have multiple smooth muscles that are joined by gap junctions such that the only innervation has to occur near one portion of this group of smooth muscle cells, that is where the varicosities are, and the rest of the communication occurs via the gap junctions. Smooth muscle excitation contraction coupling is described here. There are some similarities and some differences between smooth and skeletal muscle in terms of excitation contraction coupling. Calcium binds to calmodulin. So here we have both a similarity and a difference. Calcium is the triggering molecule for both types of contraction. In the case of skeletal muscle, calcium will bind to troponin. In the case of smooth muscle, calcium binds to calmodulin. There is no troponin in smooth muscle. In step 2, the calcium calmodulin complex will activate an enzyme known as myosin light chain kinase. This myosin light chain kinase will phosphorylate the regulatory chain of myosin, forming myosin phosphate. These steps are all different and unique to smooth muscle. Myosin phosphate will then bind to actin at a 90-degree angle. So again, we have myosin binding to actin as it does in the other forms of muscular contraction. We have a dephosphorylation step followed by a conformational shift of actin and myosin, which causes the filaments to slide. So the sliding filament portion is also similar across all forms of muscular contraction. So at this point, we've learned enough about smooth muscle contraction to know a bit more about the details of the pharmacologies that we learned earlier in terms of epinephrine, norepinephrine, and various other molecules that impact smooth muscle contraction. So for example, one can inhibit contraction of smooth muscle by activating beta-2 receptors via epinephrine. So epinephrine, when it binds to a beta-2 receptor, as we learned previously, will activate the G sub S pathway, which will increase cyclic AMP levels. Cyclic AMP levels, again, as we learned previously, will activate protein kinase A, which is a cyclic AMP dependent protein kinase. Protein kinase A will then sequester calcium back into storage, and the cyclic AMP and protein kinase A will inactivate myosin light chain kinase. The inactivation of myosin light chain kinase will lead to less myosin phosphate, and as we learned in the previous slide, myosin phosphate is part of the initiating process for muscular contraction in smooth muscle. Clinically, this is important because this is the mechanism by which beta-2 agonists work. So for example, albuterol, salmeterol, both used to treat pulmonary disease, act via this pathway. Other drugs such as theophylline and aminophylline, which are both phosphodiesterase inhibitors, also work via this pathway because by blocking phosphodiesterase, they will increase cyclic AMP levels, causing the inhibition of contraction. As we learned previously, one can stimulate contraction of vascular smooth muscle via activation of alpha-1 receptors. So for example, norepinephrine we know binds to the alpha-1 receptor. This will drive the G sub Q pathway. G sub Q, when activated, will cause phospholipase C to split PIP2 into DAG and inositol triphosphate. Inositol triphosphate will cause the release of calcium from the intracellular sarcolemma stores, and this calcium increase in the cytosol will trigger smooth muscle contraction. And this is the mechanism by which alpha-1 agonists work, such as phenylephrine. This concludes our discussion of the physiology of smooth muscle.